Neuralization begins around the end of the third week of development, when the differentiation of ectoderm forms the brain and spinal cord. Neuralization is initiated by chemical signals from the notochord. It starts with thickening of the ectoderm cells immediately above the notochord, forming a structure called the neural plate. About a year ago, I received some feedback from a student who took part 1, and they said one of their questions asked which embryonic structure becomes the spinal cord, and the answer was the neural plate. So if I were to ask you which tissue gives rise to the brain and spinal cord, you would say ectoderm. And if I were to ask you which structure gives rise to the brain and spinal cord, you would say the neural plate. The neural plate cells then differentiate into neuroectoderm. The neuroectodermal cells, represented in green at the lateral margins of the neural plate, then become neural crest cells. The thickened neural plate folds inward, forming the neural groove. As the neural groove deepens, it forms the neural folds. The neural folds gradually come closer and closer to each other in the midline and eventually fuse together. As the neural folds get closer and closer to each other, neural crest cells at the lateral margins of the neural plate detach from the folds between the surface of ectoderm and the developing neural tube. Around the same time, structures arising from mesoderm called somites begin to form on either side of the neural fold. The developing somites are depicted as somewhat red round shapes that look a little like tadpoles to me in this drawing. These neural crest cells soon divide into a cell mass that give rise to the sensory ganglion of the peripheral nervous system, specifically the dorsal root ganglia and the sympathetic ganglionic chain. Neural crest cells give rise to several other cells or tissues in the developing human, such as Schwann cells, glia cells, the meninges of the forebrain, chromaffin cells of the adrenal medulla, melanocytes in the skin, the bones of the craniofacial skeleton, and the smooth muscle that forms the middle layer of blood vessels of the face and forebrain, just to name a few structures. For the purpose of this course, we are going to focus on the ganglia that arise from neural crest cells, specifically the dorsal root ganglia. During the fourth week of development, the neural folds begin to close, forming the neural tube. I mentioned in slide 3 how the neural folds gradually come closer and closer to each other in the midline and fuse, and that the primordial somites become more defined during this stage. I see that I'm repeating myself. The only new information on this slide is that the process occurs in the fourth week. The fusion of the neural folds begins in the cervical region, and I want to stress that because sometimes students get this confused. It is the cervical region where the process of the first neural folds coming together occurs. Then the fold closures proceed cranially and caudally from the cervical region simultaneously, kind of like a pebble in the pond sort of thing. I need to stress that we're talking about a tiny, tiny microscopic embryo and that these regions I am mentioning are very close to each other, but they are distinct. The cervical region on an embryo is separate from the cranial region. Also, please be aware of the synonyms I can use to indicate the head region of an embryo. The terms cephalic, cranial, and rostral all refer to the head region of the embryo. Now, after all the folds have fused, there will be an opening on each end of the neural tube, kind of like a paper towel tube. These open ends are called neural pores, and they need to close to complete the process of neuralization. The cranial, aka cephalic, aka rostral neural pore closes around day 25, and the posterior or caudate neural pore closes around day 28. So the tail end of the neural tube closes 3 days after the head end closes. Neuralization is complete when the caudate neural pore closes. The cranial, aka cephalic, aka rostral end of the neural tube becomes the brain, and the remainder of the neural tube forms the spinal cord. This is the end goal or the main goal of neuralization, the formation of the brain and the spinal cord. Oops, I'm repeating myself again with this slide. I apologize. The cranial, aka cephalic, aka rostral end of the neural tube becomes the brain, and the remainder of the neural tube forms the spinal cord. At the end of neuralization, the cephalic, rostral, cranial end of the neural tube will have three dilations known as the primary brain vesicles: the proencephalon, or forebrain, the mesencephalon, or midbrain, and the rhomencephalon, or hindbrain. I apologize if my using the three synonyms is annoying, but it's been my experience from previous students that they missed exam questions because they weren't familiar with these terms. I promise I won't keep saying all three synonyms when referring to the head in the remainder of this PowerPoint audio. The image you see on the left is an embryo of a chicken, and believe it or not, human embryo neurotubes and somites look the same at this stage. After neuralization is complete, paraaxial mesoderm begins to organize into defined blocks of segmented tissue on each side of the closed neural tube called somites. The term periaxial is just referring to the location of the somites that arise from the mesoderm and not a specialized type of mesoderm. Somites are periaxial blocks of mesoderm. That's just a fancy way of saying somites are the circle things that appear on each side of the neural tube at this stage. In the cephalic region, these round balls of mesoderm are called somitomeres, and from the occipital region caudally toward the tail, they are called somites. As you saw in the previous slide, and where the top yellow arrows are pointing, somites hug closely to each side of the neural tube. I could also state that somites are found on each side of the notochord. Can you see the notochord just under the neural tube in this image? Eventually, each somite on each side of the neural tube will differentiate into three distinct components: the sclerotome, which will form the vertebra and ribs, the myotome, which will form most skeletal muscle in the body. Some head and neck muscles are derived from the pharyngeal arch, and finally, the dermatome, which forms the dermis of the skin. As you can see here, we can trace the origin of many tissues and structures in the body back to the mesoderm primary germ layer. The two main neurotube defects we will cover in the PowerPoint are meroencephaly, or also known as anencephaly, and the three types of spina bifida. Meroencephaly is partial absence of the brain caused by failure of the rostral neuropore to close at the end of the fourth week. If the tube doesn't close there, there's no defined place for the brain to form, specifically the cerebrum. The brainstem will form in the neural tube, and the remainder of the body can form completely to full term. Sadly, these babies are stillborn and not viable. The research I did on this condition states that it affects female babies more than male babies at a ratio of 4:1, but it did not explain why. The cause of this birth defect is still unknown. Myelomeningeal spina bifida is a fluid-filled sac containing part of the spinal cord, meninges, and nerves. It is the most serious type of spina bifida. It is typically treated surgically immediately after birth. I have even read some research experiments where the surgeon are correcting this condition in utero. Meningeal spina bifida is the next serious level of this condition. It presents as a fluid-filled sac protruding through an opening in the baby's spine, but it does not contain the spinal cord or any nervous tissue. It's just cerebral spinal fluid. This too is treated surgically immediately after birth. Is the mildest form of spina bifida. In most situations, this condition is asymptomatic. I have treated many patients over the years that did not know they had this condition until they came into my office and I took x-rays of their low back. Spina bifida results from failure of ossification of the spinous process. It presents as a small gap in the spine, but no opening or sac protruding. On this image, you can clearly see the gap between the two halves of the vertebral arch at L5. Keep in mind that vertebra bones ossify from cartilage, and cartilage is reasonably strong, so there isn't an actual space there as the cartilage appears black on X-ray. Now look a little harder. Can you see the small gap between the arches of S1? It has been my experience that if I find one anomaly on the spine, I will typically find one more. If this image was one of my patients, I would take a full spine image to rule out anomalies in other areas of the spine. Textbooks say a person with spina bifida occulta will often present with a tuft of hair over the defect area. I never saw this in my practice, and I saw a lot of spina bifida occultas on x-ray. I'm bringing this up because you cannot always count on your patients to present signs or symptoms exactly as our educational references inform us. Also, I personally remember that this was a board question when I took the boards 100 years ago.